All right, give me just a moment. I'm trying to find the folder where everything is at. All right, where are we at? All right, hi everybody. So, got a couple of things to do today. Oh, let me just put my room light on. Dum. Okay, so today we have a couple of things to do. We have a little bit of time for any questions about the mini lab report. Most of you, well, not most of you, some of you, sorry, have already submitted yours for feedback. If you haven't done so already, I strongly recommend you do so. This is due Thursday evening or Thursday night, midnight, so I can start grading them over the weekend before next week. And to be honest, the ones I've got so far have been pretty good. They're in the kind of high 70s, mid 80s kind of range in terms of grade. Usually the issue is one of like fine tuning, right? So you can knock out a pretty half decent mini lab report in not much time, but really getting up to that A grade level takes a few revisions, right? And it doesn't really matter how many revisions, to be honest, because it's just part of the process. But don't assume that the first draft that you do is going to be sufficient to get the grade that you think it deserves, right? Because they take revision, they take editing. There's quite often a matter of, no, you don't put that there, you put that there, right? If it's there, you're not going to get graded for it. It has to be there. Little things missed out, usually it's nothing particularly major in terms of the ones I've seen so far. I think 3 or 4 at least, maybe more, I can't remember, over the last few days. But the small stuff, and that small stuff adds up. Oh, I got a question about how much the lab report is worth. It's worth 200 points out of 700 for your lab. So the Excel spreadsheet was 100 points. The mini lab report is 200 points. And each lab, aside from those, and there's 8 of them, has been worth 50 points. So a total of 700 for the lab section. And that will be essentially converted into 25% of your overall course grade, right? So kind of the math is always a little awkward, but it's nothing you can't fix with a calculator. So it's definitely something that you would want to do as well as you can, because it's worth a big chunk of your lab grade. And your lab grade really is how one way, not the only, but one way in which you can get a good course grade, particularly if the exams are proving difficult, right? So, and the ones that have done so so far, obviously, I'm not going to tell them what grade they got, right? But there's certainly the subsequent revisions are definitely more polished and will be graded more highly than their initial submission, right? So I will be, what am I doing tomorrow? I'm actually going to be out and about tomorrow. So if you want feedback on this, then on your mini lab report, I'll most likely be able to get to those tomorrow evening when I get back from we're going out for a little day trip with the family. So don't leave it too late, otherwise I won't have time to get it back to you in order for you to submit any new revisions. Oh, thank you, Andrew. It's part of the job. And also it's part of how you get better at doing this, right? I can't emphasize this strongly enough. I can't think of any single piece of written work that I've ever produced within the last, I don't know, 20 years or so that has not gone through multiple rounds of revision. Typically, my boss at the time or my PhD supervisor were the ones, hang on a sec, Austin, who gave me that feedback, but now I get it from my peers, like my colleagues and from people who review papers. So the end result is always better than the beginning. That's a guarantee, and that's just part of the process. So I got a question about the Excel spreadsheet. That was actually due last, oh shoot, when was that due? Last Thursday, I think. But because I'm a kind-hearted soul, if you get it to me today, I'll grade it today. But yeah, it's really important to pay attention to the deadlines in the summer schedule. Where is it at? This isn't perfect because sometimes it says beginning of the lab, but it gives you an idea of the date. So for example, wherever it says Excel spreadsheet with cross date due beginning of next lab, that means it's due on Thursday, right? So I would, again, strongly recommend putting those dates into your whatever calendar app or whatever you use. I use the calendar in Outlook. It tells me everything, even when my dad's birthday is. And reminds me incessantly until, you know, it is his birthday. Put stuff in there, you will then not forget. And I mean, I'm fairly accommodating when it comes to miss stuff, but others may not be as much. So I wouldn't get too used to it. But yeah, if you can get that spreadsheet to me by today, I'll grade it as if it was handed on time. But the lab report, because it's a bunch more work to grade those, typically going to be less accommodating for late stuff because, you know, other things will be happening that need my attention after that. Any other questions on the mini lab report? Nope. Cool. All right. So essentially what I want to do now is just go over a little bit of the background behind what we're going to be doing next. So I shouldn't have dropped that. Let's go back to this again. So essentially this is from today onwards until the last day of classes, right, which is 28th of July, which is really not that long away, to be honest, which is kind of crazy. Is going to be what would have been a whole semester long fly project. And so the first part of that project is the linkage analysis, which this mini lab report is finishing up that work. And that really deals with the kind of more Mendelian genetics and statistics aspect of it. But we've also, as part of that project, we also have a kind of more molecular biology, bioinformatics type project, right? Which are also really important skills that you'll need to learn to, you know, succeed and go into the next stage of your degree or your career. And essentially what this is doing is kind of recapitulating or walking you through the process of figuring out what causes a particular phenotype. So I haven't done this because, you know, most of this work was done before I got into the worm field. But an awful lot of researchers' work was to do a mutagenesis screen, so get a bunch of animals, worms in this case, mutagenize the crap out of them, and look for phenotypes of interest, whatever that may be. So for one of my postdoc bosses at Penn State, her project was to look for vulval development phenotypes, right? Because vulval development is a kind of a developmental paradigm in C. elegans biology. And so she just, you know, mutagenized millions of worms. Most of them died, obviously, because that's bad for them. And then she got a bunch which had interesting phenotypes. And so her job was to figure out, well, what causes that at a molecular level? And this is essentially the same process that you would take if you were doing a similar project, right? And it's an important kind of approach to take, right? Because it's a very classical approach to a problem, which is called forward genetics. So you don't know what is involved in something. You just screw things up and see what produces the phenotype that you would predict, right? But even once you get that, you have to then go back and say, well, that's all well and good, but what's causing this phenotype, right? What gene has been affected, or genes, right? And that comes back to that example of complementation that we talked about in lecture. What pathways or processes are involved in the normal function of whatever, right? What the function that you've screwed up during your mutagenesis screen. And so essentially you go through those one by one, and that's exactly what, you know, on computer, unfortunately, we'll be doing with this project. And so we have a phenotype, which is the yellow phenotype. Sorry, I keep looking down at the bottom of the screen. It's at the top. Where is it? Here we go. And so this is written the way it is right now, because this is actually these are some supplementary materials for a paper that Dr. Page and I are hoping to submit this summer about this particular project for other genetics teachers to use. And so essentially we have, where we are, this phenotype here. This is the yellow phenotype. So you've already come across it in the linkage experiment, and that's good. But essentially we picked this one because it's got a very well-characterized mutation, which is the one that we're going to be looking at, which results in the yellow phenotype. Now, we don't actually fully understand the molecular basis of the yellow phenotype. We know what sequence change has occurred, but we don't fully understand what that protein does normally. It's partly involved in pigment production, as you would expect, because these are basically like the pasty white Brits of the fly world, and these are the suntanned central Texans, let's say. So these are brown. You can see the darker stripes on their abdomen. And when you look at these, they're a lot paler all over, right? So the body's paler and the stripes are paler. The yellow gene does do an awful lot of other things as well. So exactly how that reduction in pigmentation happens, we don't actually fully know, even now, right? And this has been studied for decades. Okay, so essentially, we're going to start with these flies virtually. And what you would normally be doing in this lab would be kind of having a closer look at different fly aspects. You know, these are females because they have these bigger abdomens. These are male because they're smaller and have a solid band at the bottom. Males also have what are called sex combs, not entirely sure what they do. I think they're for like holding on, yeehaw, to the ladies. I think, not 100% sure. That's my own personal interpretation. And essentially, you'd spend time practicing some of the fly managing stuff, right? Microscopy and the like. Obviously, we can't do that. So really, this is just a quick overview of what you'll be doing for the next few weeks. And so essentially, the idea is to go through all those steps. So the first one would be, what's your phenotype? Okay, and you always compare that phenotype to wild type. So this is our phenotype of interest, the yellow body. This is our wild type version, which has a brown body, right? You always have to compare it to wild type, because sometimes stuff just looks weird just because it looks weird, not because it's actually been messed around with. Then what we're going to do is go through all the different steps. So the first step would be, if we want to look at the genetics of this mutation, we need DNA, right? Because that's what it's our kind of base material. So we'll be doing a virtual DNA extraction, right? So kind of covering the principles of DNA extraction and the like. Then you're going to be doing a virtual PCR. And again, my apologies for you not being able to do this in person. Obviously, you lose a little bit of the experience, but hopefully you get the knowledge and the analytical skills, right? Do a PCR, so we need to use a technique called polymerase chain reaction. Yeah, I know. I'm sorry, Alicia. It is a pain. There's not much we can do about that, unfortunately. Or I mean, you could buy like your own PCR machine and a bunch of stuff if you've got a whole bunch of money hanging around. They're only a few $1,000s each, like secondhand. It's super cheap. Yeah, but hopefully, I'm not sure about micro. Micro is more about isolation. Hopefully, at some later point, one of the other courses that you'll be taking, you'll get a chance to do this. But anyway, the aim of PCR is to really amplify a target sequence into like loads of it, so you can actually do something with it. And then we'll clean it up. Again, the principles behind that. Send it off for sequencing. We have a whole folder which should be in one of the folders. Let me just double check. Where is it at? Yeah, this will be in the last folder, which is for module 6. Yeah, there we go. It's a zip file with, I can't remember how many sequences, 20 or so, I think, which are actual sequences from doing this. And then we'll finish up with a lab using a program called Eugene, which you'll need to download and install on whatever device you're using to do this work. I'd recommend at least a tablet, if not a laptop or computer of some sort. And then we'll be actually using that software to analyze those sequences. And so Eugene is a hugely powerful and not super user-friendly program, but it's also very much like any other that you might use to do any kind of bioinformatics work in a molecular biology lab. So essentially, even though you're not going to be doing like the wet work, as it's called, the bench work, doing the PCR and grinding up flies and things like that, you'll still be doing every single step in terms of your knowledge and skills as you would be doing if you were doing this for real. And then the aim is to essentially end up at the end in the Eugene lab with a definitive answer as to why the yellow flies that we sequenced are yellow in terms of the molecular genetics of that gene. And that's pretty much it. So there's not a huge amount else here. This is really just how to wrangle flies, stop them flying around and annoying other people, and how to phenotype them, right? As well as how to use a stereo microscope. Most of you know how to use one already, so that's not too much of a loss. And then, so there's nothing due for next lab on Thursday for this. The only thing due on Thursday is the mini lab report. And there will be, after Thursday's lab, like the same, a little series of questions or things you need to do on Blackboard for the DNA extraction assignment. And that will be the same for the rest of it. I think there's another couple that I need to do. DNA and I think maybe PCR, I can't remember. But those will be up in good time. And that's pretty much it. So, you know, if you're interested in genetics or molecular genetics or molecular biology, any of that kind of stuff, kind of consider this like a dry run, essentially, through all the different steps. So that when you do get to do it for real, in whatever context that may be, you'll already be familiar with, you know, oh, this is what I need to do. And I need to do that for that reason, because blah, you know, that's kind of the hope. And that's pretty much it, really. I could keep on talking for a while, but I think you've probably heard enough of that already today. Does anybody have any questions about anything in particular? Hopefully the Zoom experience has been a little bit more straightforward for people than the WebEx one. If you have any particular feedback towards the end of the semester about Zoom versus WebEx, feel free to send it to me and I'll pass it on to the IT person who's in charge of this trial. Because it would be, certainly some classes are going to be online in the fall, whatever happens. So it would be good to get Zoom more widely used rather than WebEx. So if you have any feedback for that, please pass it on. Cool. All right. Well, that's pretty much it from me. I'm going to let you get off so you can get going on the exam or, you know, whatever it is that you have left to do today. And I shall see you on Thursday, where we dive deep into transcription. Cool. All right, everybody. Take care.